Partch. I'm an assistant professor in the Department of Chemistry and Biochemistry at UC Santa Cruz. I would define our approach to circadian rhythms as trying to get at the molecular basis of circadian rhythmicity. That is to say, how do the clock proteins work together to create 24-hour molecular clocks? And to do that, we really take a biochemical approach. And one of the ways that we approach that is with structural biology, which lets us essentially visualize or see the proteins interacting. So for example, I have an undergrad in the lab who is trying to do some biochemistry on a region of the protein clock, famous exon 19. It's a little piece of the clock protein that if you don't have it, you don't have a circadian rhythm. But we don't really know what it does yet. So I have an undergraduate who's been studying the biochemistry and the structure of it. So mostly in the lab, we're focused on mammalian circadian rhythms. We're interested in how animals measure time. But just recently, we started a collaboration with Andy Lee Wong and Susan Golden to look at the cyanobacterial circadian clock, which is just a really exciting clock where you can put 3 purified proteins in a test tube and basically watch them tell time. I would say we're really true in vitro people, and so we actually, at this point, we don't work with live mice or live cyanobacteria. Instead, we purify the proteins, we make recombinant proteins. So we use bacteria or insect cell systems to express the proteins that we're interested in, and then we just go to town mixing them and playing with them basically in test tubes. So we study cyanobacteria and animal clocks, but we really work with the proteins themselves. I really love thinking about the intersection between biology and sort of quantitative aspects of biochemistry. I'm like addicted to data. And so the ultimate goal that we have is to build enough understanding about the biochemistry that we could really make the leap to create therapies that would control circadian rhythms.